Well, good morning, good afternoon, everybody, and welcome in with another very exciting Exploring by the Seat of Your Pants broadcast. If you're joining us for the first time, we are all about bringing conservation, adventure, and science into classrooms around the world, and you're catching us in a very special day before the weekend because we are joined back live in our epic Gairdner Foundation series. The Gairdner Foundation showcases and celebrates the best biomedical researchers in the world. Many of their laureates have gone on to win the Nobel Prize, and we like featuring the most cutting-edge science working to understand the world and save lives within it in this epic series. You've got another program coming up at the end of May with Rodolphe Berengu, so do stay tuned for that, and some epic daily dives coming up with all sorts of great laureates. But check out Gairdner.org to learn more and do a deep dive in on scientific adventure. Now, as we dive into today's broadcast with the most featured speaker we have had in this series in our history, I want to note, Lewis, I'm going to bring you in. Lewis joining us from the University of Toronto. Welcome back in, Lewis. Now, Lewis and I did an incredible, everything that could possibly tech wrong happen in a broadcast happened in advance of this, but I'm so glad that we got to make it happen and go live, even if it's a little haphazard. I'm going to be sharing the broadcast from my side, but Lewis, it's so nice to have you back. We did it together, and I know you got a lot to share with us, so I'll leave you to take us away in just a second.
Sounds great.
Thank you so
All
much.
I need is the first slide, which is taking its time, I guess, to appear. But as it does, I just want to tell you that science is all about persistence. So we've had problems with this, getting this up on the screen here, and I have no control over what I do. Scientists like to have control over what we do. We like to do experiments. They're carefully controlled experiments so that we can learn things about whatever system that we're studying. But this is very hard to do in this present broadcast because I have no control. So I want to begin in a sort of a haphazard way. I'm sorry that I can't point to things, but I want to tell you a little bit about science. And science is a lot of different things to a lot of different people. Science can be physics, where we try to understand the nature of materials and how they interact with one another at the most fundamental of levels. Science can be chemistry, where we're interested in molecules, how molecules interact with one another. Science can be biology, where we want to understand how organisms live and die and how diseases and mutations affects the organisms. So there's all sorts of different things that we can study when we study science, and it just really depends on what your interests are. If I could have the next slide. So just to give you a little bit of background about what science is all about, astronomy is the study of outer space. So that's really a branch of physics where we look to understand the forces of the universe. How did the universe evolve? Physics is dealing with molecules in terms of the fundamental forces involved and trying to understand the nature of the world around us. Chemistry deals with matter. Matter is the stuff that stuff is made of. How does matter combine with one another? We come up with various ways of, for example, developing vaccines using the molecules of life, the chemistry of life that has really profound implications on society. Medicine, of course, we're interested in how to keep our bodies healthy. Psychology is the science of our emotions and our feelings. Biology, I've spoken a little bit about. Genetics, which is a branch of biology, to understand how the molecules of life can confer the various properties of living systems. Understanding DNA, which is the blueprint of life, how that translates into all of the different molecules that play a role in healthy organisms. Computer science, of course, is the science of computers. And if I was a little bit better as a computer scientist, then maybe I'd be able to point to my screen and actually tell you about what more specifically I'm trying to do. And paleontology deals with the past, fossils, dinosaurs, and ancient forms of life. Could I have the next slide, please? As a scientist, we often deal with mathematics, and this is just a bunch of gobbledygook, various symbols. You're probably all familiar with E equals mc squared, even though you're 10 and 11 years old, I'm told. So as scientists, physical scientists, and I am a physical scientist, I have to describe the universe in terms of equations. And for me, the universe is going to be molecules. Next slide, please. So I want to just focus on two branches of science. One is physics and one is chemistry. I'm actually at the interface of chemistry and physics. I'm called a chemical physicist. And here we have a physicist. He's got this white hair. He may even be Isaac Newton. And you can see that an apple is falling. Actually, I guess about 4 apples have already fallen. And Isaac Newton has a problem. He wants to understand what's going on. Next slide, please. And so what Isaac Newton is going to do is he's going to use the scientific method. He's made an observation, and that is that apples fall down from trees. They don't go up from trees into the oblivion, they fall to the ground. And so the question is, why do apples, in fact, fall to the ground? What are the forces that are involved, say, between the apple and the earth that causes the apple to fall as opposed to rise? So he makes a hypothesis. Well, maybe there are forces between them, and maybe the forces depend on the masses of the apple and the earth. And maybe they also depend in some way on the distance. And then he does an experiment. He drops apples from various distances, and then he analyzes all his results, and he comes up with a really important conclusion, and that is shown on the next slide, please. So this conclusion is shown with the equation, which I would normally point to. Isaac Newton has all of a sudden become rather young. He no longer has white hair. He has brown hair. And he comes up with an equation that relates the force between the apple and the ground. That's as the apple is falling, and it depends both on the mass of the apple, the mass of the ground, and the distance between the apple and the center of the earth. Next slide, please. So that's an example in the world of physics where we try to understand things mathematically. Often we do so also in chemistry. So here I have an atom, which is sort of the fundamental building block of much what I'm going to talk to you about. Of course, atoms are made up of electrons. These electrons are the green things around the circular nucleus, okay? And the nucleus contains particles like neutrons and protons, and they're in turn made of other particles. And as a chemist, I'm interested in electrons, neutrons, and protons. They confer various properties to matter, and I want to use those properties to study my molecules. Next slide, please. Now, in chemistry, we have very beautiful elements. So there's roughly 118 elements. If you take pure atoms and you put them together, you get elements. So here we have gold in the upper left-hand side of the screen. If you own a lot of gold today, you are going to be very, very wealthy. So gold is a beautiful element to have in your back pocket. But there are other pretty elements. For example, going to the left-hand bottom side, we have copper. Copper is really important for transmission of electron signals. So you have basically currents that are transmitted, which is really important in electronic circuits. Sodium is really interesting. It reacts, it combusts with oxygen to give the beautiful flame, the yellow flame shown in the bottom right-hand side. Next slide, please. So we have atoms, as I told you about, and these atoms are going to be the building blocks of molecules. And in the bottom half of the circle here, we have DNA molecules. And DNA molecules are really important because DNA molecules are the blueprint of life. They code for the proteins in your body, and the proteins are the movers and shakers. The proteins are the molecules that are going to carry out function, and we want to understand that function. And I'll show you a little bit about that later on. Next slide, please. Now, chemistry is absolutely awesome. Those of you who don't believe me, well, try chocolate. Do you like chocolate? If you like chocolate, chocolate has an important compound in it, and that's shown in the top left-hand side. Caffeine, if you drink coffee, you're too young to do that. Well, that looks sort of like chocolate with just a touch of a difference. It's just an extra carbon, actually what we call a methyl group is attached, and that generates new characteristics. Happiness, serotonin, is another compound that's important. Alcohol will make you drunk, and you can see that one. Love, that's dopamine. Adrenaline is the sort of thing that you get when a tiger is chasing you and you've got to run really fast. So these molecules in chemistry are super important for giving us the various properties that we call happiness and love and adrenaline and chocolate. Chocolate isn't a property. Chocolate is, when we eat it, we feel good. So that's a property. So next slide, please. And our human body is made up of, not chocolate, of course, but various elements. For example, oxygen. 65% of our body is oxygen. We have water in our body, a lot of water in our body. That's 2 hydrogens and an oxygen. That's where that much of the oxygen comes from. We have carbon, we have nitrogen. So we have various elements that make up the human body. So to understand how the human body works at a detailed level that involves molecules, we have to understand something about the elements. Next slide, please. Now, science is easy, and I want to illustrate that to you. But actually, learning the rules of science can be fairly complicated. Just like anything in life, you've got to learn the rules before you can play the game. Take football. Now, in football, in the National Football League, there's roughly 30 run plays in an average playbook and 200 pass plays. And there's roughly 5 or 6 or maybe 10 variations on each play. So that makes over 1,000 plays. And as a football player, if you're earning, $2 million a year, you've got to execute those plays at top speed, knowing that at some stage, somebody is going to smush you. And that somebody weighs 300 pounds and is made of steel. So that's an example of something that is really hard, even though you get paid well to do it. By contrast, science is much easier than playing football in the National Football League. Next slide, please. And the reason that it's easier is because there's a lot fewer elements. There's just 120 elements or so. So we don't have to worry about such complications like learning 1,000 plays. Now, scientists are interested in patterns. And I want to give you an example of one such pattern that we're interested in. Let's suppose that you have 10 numbers, 1, 2, 3, 4, 5, all the way to 10. And your teacher says, well, what's the sum of the first 10 numbers? Well, the way that you would do it is probably 1 plus 2, that's 3, plus 3 is 6, plus 4 is 10, and so forth. But in fact, you could be a little bit sneaky. You could look for a pattern like what a scientist would do. And what a scientist would do if he had to sum, or she had to sum the first 10 numbers, he or she might take the number 1 and add it to the last number. So the first and the last number, that gives you 11. Then take the second number, that's 2, and the second last number, add them, and you get 11. Then take the 3 and the 8, you get 11, and so forth. And you're going to get 5 11s out of this that you have to sum, and that's 55. That's what the sum of the first 10 numbers is. Now, the sum of the first 100 numbers, you do the same thing. 1 plus 100 is 101. Then take the second number on the left and the second number on the right. That's 2 plus 99 is 101. 3 plus 98 is 101. And you're going to end up with 55 101s. So that's 5,050. That's the sum of the first 100 numbers. Dead simple. But it's simple because we noted a pattern. And that's what scientists try to do. We try to find patterns which provide us a way that we can understand the properties. In this particular case, the property happens to be the sum of the first 10 or 100 numbers. Next slide, please. Now, this always fascinated me with mathematics. And I wanted to look at something that combines sort of math, chemistry, and physics. But what I want to do now is I want to illustrate that scientists can look at very tiny things, which is what I do, like molecules that you can't see without the help of really, really powerful experiments. Or we can look at really, really big things that are far away that you need, for example, telescopes. So let's zoom in. On the right-hand side, we have a couple having a picnic. And what I'm going to do is I'm going to zoom out from the couple. I'm going to get farther and farther away from them. And then I'm going to get closer and closer to them. And I want to show you the differences in the length scales, which sort of describes the differences in the sorts of sciences that you can study. So let's begin with the right-hand side. I'm focusing on the guy now, and I'm about, let's say, 1 meter away from his shirt. Next slide, please. Now what I'm going to do is go farther. I'm going to go farther above. So I could be 10 meters away. That's on the right-hand side. Then 100 meters away in the center, and then 1,000 meters away on the right-hand side, on the left-hand side. And as I go further and further away, you can see that the couple get further away, and I don't see them anymore. I begin to see more and more of the surrounding area. Let's keep going. Let's go further and further. Now I'm about 6 miles away on the right-hand side, and now I'm about 10 or 100 kilometers. I can't read that. I guess it's 100 kilometers away on the left side. So I'm getting further and further away. Okay, let's continue on. Now I'm further away on the right-hand side, and I can see the Great Lake that is close to where the couple are just enjoying themselves in one little corner. And as I go further away on the left-hand side, now thousands of kilometers away, what's that thing in the middle? Well, we call it Earth. So now I see the planet Earth. And now I'm going to go further away in the next slide. And now on the right-hand side, I see the solar system, okay? And then I have the Milky Way galaxy in the middle. And then as I go further and further away, this is 1 billion light years away. So that means that light, which is moving really quick, it would take 1 billion human years to get from where I am now back to that couple that are lying down. Next slide, please. Okay, now let's go back to them and let's take a deep dive inside. Next slide, please. So now as I go inside, you can see I focus on the hand. I get closer to the hand. As I move from left to right, I'm going inside. So you can see now that you have the indentations, the handprints, if you like. Then I go inside into the blood vessels. Next slide, please. I can begin to see a white blood cell, which is inside the blood vessel. And then as I go, that's on the left-hand side. As I go to the right-hand side, I can see the molecules inside that white blood cell. This is DNA. This is the blueprint for the white blood cell. Let's take a deeper dive inside. On the left side, now we see the individual strands of the DNA. And as I focus more deeply, you can see the individual molecules on the right-hand side. Next slide. And then I can begin to go inside the atom. I'm beginning to see the nucleus in the middle. And if I go down where I have written atom structure, you can see the red and the blue are the protons and the neutrons of the nucleus. Next slide, please. So the question that you have to ask yourself once you decide to become a scientist is, at what level do you want to understand the world? What is going to satisfy you? Do you want to understand the overarching principles, or do you want to dig really deep and look at the tiny little atoms, or do you want to look at the universe as a whole and look at the fundamental features of the universe? We're always interested in understanding the principles, but is it the principles that govern the behavior of tiny things, or of big things, or of things in between? All sorts of different technologies have been developed to address specific questions. Now, for me, it's understanding biophysics. I want to understand the molecules of life. These molecules are going to be really, really tiny, but at the same time, I want to understand these really tiny molecules that are related to biology and biological function. I want to understand them with the principles of chemistry and physics. So I want to be a physicist on the one hand and a biologist on the other hand. I want to combine the two. And often in science, at the crossroads between two different areas of pursuit, whether it's biology and chemistry or biology and physics, there's a very rich intersection that allows you to make rapid progress using technologies from one field and applying them to another field. Next slide, please. So when I was your age, I wanted to do something really, really important. I didn't really know what it was. I still don't know what it is because I haven't gotten there. But I figured curing cancer would be an important thing. And I bet that if you were to ask a lot of budding scientists who are 10 and 11 years old, they would say, well, I want to cure cancer. That's easier said than done. But I wanted to combine math with curing cancer. You see, I've always been interested in math. I've had this fascination in numbers, in patterns, in using quantitative relationships to understand other things. So I want to be like this lady on the right, who's a mathematician, and the gentleman on the left, who of course is a cancer researcher. Next slide, please. And I haven't figured out how to combine cancer research with math, but what I want to do today is to tell you a little bit about my path, because I think I've figured out more or less my path, at least up to now. But of course, a path always evolves. So there could be some interesting experiences going into the future. In fact, there will be. But I'll tell you about my path to this point. Next slide, please. I want to combine my love of math and physics, shown on the left-hand side, with understanding how stuff works in the cells. Now, a cell is basically an entity. There's a biological membrane that separates the inside from the outside of a cell. And inside the cell, there's a lot of molecules. And these molecules are densely packed, super densely packed. It's like the richest soup that you can ever imagine. The potatoes and the meat and the peas are all touching one another. And somehow everything is organized and life happens. So the question is, how does that occur? How can I use the laws of physics to understand the laws of biology? Next slide, please. And before I can really tell you what I do, I have to describe the movers and the shakers inside of a cell. Next slide, please. And the global picture is as follows. So we have these things called cells, and these cells contain a nucleus, and the nucleus contains the blueprint. That's basically the genome. That's the chromosomes, the DNA, which codes for various proteins. So we have a blueprint. The blueprint is a code. The code is going to tell us how to construct the movers and the shakers inside our body, which are called proteins. These proteins have different shapes, and the shapes of a given protein are in fact going to change as the protein interacts with other proteins. Just like if you were to give your friend a hug, both of your shapes would change a little bit. Well, the same thing if a protein hugs another protein. So we want to understand the shape of these proteins, how the shape changes as the proteins hug each other, and then how it is that disease mutations will change the shape of the proteins and therefore change the way that these proteins hug each other, because that can invoke disease. Next slide, please. Now, don't get confused. When we talk about proteins in everyday life, like if your mom says, eat your proteins, she really means eat your eggs or eat your cheese or, finish your steak or your fish or drink your milk or something like that. The food groups are called proteins. But when I talk about proteins, I mean the little incy-bincy molecules that are inside a human body. That's the proteins that I'm talking about. Of course, they're in steak and cheese and fish, etc. as well. But I want to talk about not beefsteak, but rather about tiny little molecules that are made up of atoms. Next slide, please. Now, proteins are super important, and I illustrate that on the right-hand side of the slide, where you can see this lady here, okay? And she's basically saying, well, what do proteins do? Well, proteins are important because antibodies are proteins. What are antibodies? Antibodies are proteins that protects your body against bacteria and viruses. So if you get a shot, for example, a COVID-19 shot, that generates antibodies, and these antibodies are going to protect against the COVID-19 virus. Proteins can be cellular messengers. As information is transferred from, say, outside of a cell, inside a cell, there has to be a messenger which allows that information to get through. Because remember, there's this giant gate, which is called the biological membrane. You all have muscles. Well, muscles are made up of proteins, muscle proteins, that are really important if you want to walk and run and make a fist and move your arms and legs. Your hair and nails are made up of proteins. Your blood has really a very, very important protein called hemoglobin. Hemoglobin carries oxygen to various parts of your body, and this is super important. You would not be able to live without oxygen. You all know that. Hemoglobin is the carrier of the oxygen to the various parts of your body that you need to get oxygenated. Your brain and nerves are made up of ion channels. Ion channels basically are important for allowing controlling signals. Basically, we have ions. Ions are like sodium ions or calcium ions. These ions are going to essentially be important for transmitting an electrical impulse from one nerve to the next. And these impulses are going to require ion channels to open up and close to transfer the information from one nerve cell to another. Enzymes are essentially proteins as well. And these enzymes are going to carry out reactions. So inside your body, there's a whole bunch of different things happening, a whole bunch of different reactions. And a lot of them without enzymes would take hundreds of years to happen. Well, we don't have hundreds of years. But with the enzymes, we're able to speed up the reactions to the point where they can happen literally in seconds or milliseconds, really fast. That's important for everything that we know about life. Also, proteins can be construction workers. They can be front-end loaders, the bulldozers that essentially build up various parts of the cell, but also destroy it when these parts are essentially damaged or no longer required for use. Next slide, please. And so you can see that going from the blueprint of life, the DNA on the left-hand side, we generate proteins. Proteins do a lot of the work, perhaps not all of it, but they do a lot of the work in the cell. Next slide, please. And so we would like to be able to understand these proteins, these shapes of these proteins. Now, remember that these proteins are three-dimensional, but I can't show you things in three-dimensional space today. I can only work with two-dimensional space. So you have to understand that what I'm showing here is a bunch of protein molecules that are squished down from 3 dimensions into 2 dimensions. And you can see that they're a bunch of different shapes. And the shapes are really important because they confer the properties, the biological properties to these molecules. Next slide, please. Now, proteins are made up of building blocks. These building blocks are called amino acids, but I'm just going to call them beads. So you can imagine that a protein is like having beads on a string. And there are 20 different colors of these beads because there are 20 different amino acids. And here I show on the left-hand side, you have a string and you have beads on the string and you have a bunch of different colors. Imagine that you have 20 different colors, and imagine moreover that certain colors attract each other. So perhaps the green is attracted to the yellow, but not to the red. So greens want to be close to yellows, but they don't want to be close to red. And reds might want to be close to blues, but not close to some other color like green. And so you can imagine that that creates a particular relationship, a three-dimensional relationship that brings the beads that are attractive to each other close by and the beads that are not attractive further away. That gives a shape to the protein molecule, and many protein molecules have a defined three-dimensional shape, but not all of them. Some protein molecules are like plates of spaghetti. They're all over the place. And it's like one strand of spaghetti that interconverts with another strand and then with another strand. So they assume a whole bunch of different shapes essentially at the same time. And as a consequence, it's not a defined three-dimensional shape, but it turns out that this ensemble of different shapes, this dance between different shapes, is absolutely critical for the function. Next slide, please. So let me just show you a few proteins. On the left-hand side is a protein called calmodulin. And you can see that there are 4 black dots. These are little ions that bind to calmodulin. And calmodulin is really important for carrying out signals in the body. And some of the signals require that calmodulin binds with target protein. So it has to give a hug to other protein molecules. What I show on the right-hand side is a picture of the calmodulin on the left-hand side, which is bound to a red molecule. The thing in the middle that looks like a helix that is red is another protein molecule. And as the two molecules hug each other, you can see that there's a big change in the shape of calmodulin. And that often happens, that when we get a molecular hug, there's a different shape, and that confers a particular functionality, in this particular case, to the calmodulin molecule. Next slide, please. Here we have an antibody molecule. Sort of looks like a stick upon which there are balloons, but I want to focus your attention on the green and the yellows. The green and the yellows of this antibody, which is a protein molecule, are going to recognize foreign invaders and neutralize them, essentially take them out of the picture so that we don't get sick. So these greens and yellows are super important, and they recognize foreign invaders. You have different greens and yellows, if you like, for different invaders that interact with them and essentially prevent them from carrying out their nasty thing that they'd like to do. Next slide, please. You have some proteins which are engines. So what I show at the top part of the slide is a car engine. Well, proteins are engines. They're not car engines. They're microscopic, tiny engines. What I show on the top is a macroscopic engine of a machine, but these tiny engines work by moving parts as well. Proteins, a couple of different engines are shown in A and B. In A, proteins can print money, but money in the cellular world is not a loonie or a toonie, but is something called ATP, another molecule that has a lot of energy in it. So what we're going to use is we're going to use proteins that are going to produce money, which are tiny molecules called ATP that have a lot of energy in it. And that energy can be used to allow various reactions to occur, to get the reaction over the hump, to give the reaction a push. That's what money can do. Money can buy you things, in this case, can cause reactions to occur. Now, also, these molecular machines can be very useful because they can be giant steering wheels. They steer cells. So suppose we have a bacterium, and the bacterium, this is in B, contains a particular machine that steers it towards food, but away from harm. For example, another bacteria that wants to eat it up. So you can have large machines that are either printing money or that are steering cells from harm's way or towards food. Next slide, please. You can have proteins that serve as gates. So remember that I mentioned that cells are sequestered from the rest of the world through a biological membrane, which separates the inside of the cell from the outside of the cell. But information has to be transported from outside to inside, and this occurs with the help of ion channels. So what I show here, the left channel is closed. Where it says closed, there's a particular shape of the molecule that prevents these little yellow messages from going inside the cell. But when we open the channel, now these yellow messages can go inside the cell. So these molecular entities, these molecules, can serve as gates. Molecules can also read messages and follow instructions. For example, on the left-hand side, I have a big molecule called a ribosome, which is going to read ultimately the code from the DNA in a different form, as it turns out, called RNA, but those details aren't important. The point is that it translates the genetics, the genetic code, the DNA into protein molecules that then carry out biological function. Next slide, please. Some proteins serve as garbage cans. We all know that garbage cans are pretty important. You wouldn't want to keep all of your garbage inside your house. Well, you don't want to keep the garbage proteins inside the cells neither. If you keep the garbage proteins in the cell, what can happen? Well, these proteins can clump together and they can cause neurodegenerative diseases, for example. Or they may carry out functions that the cell doesn't want to carry out, and that leads to, for example, cancers. And so the cell has a giant garbage can, and that's shown on the right-hand side. It's called the proteasome, and it's a machine. It's made up of movable parts, just like the machine in your car engine is, except that it happens to be really tiny, although I've drawn it really, really big here. Everything is relative. If you look at the small molecule on the left, in the left corner, the lower left-hand corner, that's another protein. It's the first protein that I showed you in the series today, but it's much smaller than this big garbage can. Of course, this garbage can has to fit in a whole bunch of different proteins so that it can, in the case of the proteasome, chop them up into little pieces and get rid of them. Next slide, please. Now, proteins are not static. All of the pictures that I've shown you are of something that is highly static. It doesn't move. And that's just not the way that these things work. So we can't capture function, which is what I want to do. I want to understand how these proteins work. We can't capture the function in just a single picture. So what I try to understand is how the pictures change in time, or as I like to call it, the dance of life. And why does this matter? Let me illustrate. Next slide, please. So I have a car engine in the top left-hand corner. And if you want to understand how the car engine works, which is pretty important when your car engine doesn't work, you're going to have to be able to fix it. You can only fix it if you understand something about the engine. You have to understand something about how the parts move with respect to one another. And it's the same thing that holds on the bottom right-hand side, where I show this giant garbage can where you have movable parts as well. Next slide, please. Now, let me try to illustrate why we need to have a bunch of snapshots and not just one. And I'm going to do that by showing you pictures of babies. So suppose that you existed in a world where you've never seen a baby before. And now I show you this slide here. Well, now you've seen, in fact, two babies, and you can make some conclusions. You can conclude that babies are always happy, as you can see in the upper right baby. And babies know how to feed themselves, and babies have green and white paraphernalia. We call them clothes. And you can learn something about babies. But if we go to the next slide, you quickly realize that babies perhaps don't do a very good job eating. Here we have a baby that's not wearing any clothes, a good thing too, that's doing a better job getting food on her than in her. Next slide, please. And the baby eventually gets a little bit frustrated and begins to cry. The baby wants to eat. The baby's having trouble eating. So not all babies are able to feed themselves too well. Next slide, please. So if you want to understand what fundamentally makes up a baby, you have to look at all 5 slides, all 5 pictures. In fact, you have to look at a lot more pictures than that to understand the full complement of what it means to be a baby. You have to understand how babies respond to their environment. So too, if we want to understand how molecules work, in particular the dance of life, we have to understand how the molecules of life respond to their environments. What happens when a molecule of life gets spaghetti spilt all over it, or can't feed itself, or things like that. Of course, I'm being a little bit silly, but what happens when a molecule of life is diseased? It has a mutation that gives cancer. How does it behave then relative to a molecule that is working properly, that is a healthy molecule? Next slide, please. What I'd like to show you now is just that molecules move about, and that we, just like we need to understand multiple pictures of the babies to understand what a baby is, so too we need to understand the multiple pictures and snapshots of a protein. I don't know, Jesse, if you can, there's a little button there below the red that if you hit, you'll start yes, there it is, if you hit that. Okay, so what we have in blue is a molecule that is going to be acted on by another molecule in red. The molecule in red is an enzyme. It speeds up reactions. And what this red molecule is going to do is it's going to snip the blue molecule into 2 parts. The blue molecule by itself, that is to say without the red, would never be able to do that. But with the red, you can chop the molecule in half. So this is an example of how the change in shape of the red molecule, as it encounters the blue molecule, allows a chemical reaction to occur. In this case, chopping blue in 2. Next slide, please. Now, I want to understand molecular dances using magnetism. Well, why do I choose magnetism? What even is magnetism? Next slide, please. Now, let me just tell you a little bit about magnets. I think many of you played with magnets before, and you know that a magnet has a north pole and a south pole. And moreover, you know that the north pole of a magnet is attracted to the south pole. If you take 2 magnets and you orient them in such a way, they will attract each other. But if you flip one of the magnets 180 degrees so that the 2 north poles are now close, they will repel each other. And the same if the 2 south poles were close. You're going to get repulsion. So if you have north and south that are close, you get attraction. Next slide, please. Now, here's the thing. I work with molecules. The molecules that I work with are protein molecules, the movers and shakers in the cell. These protein molecules are made up of atoms. They're made up of beads, as I said, but each bead is made up of many, many atoms. And these atoms contain nuclei. And inside the nuclei, there are little bar magnets. Okay, so inside my protein molecules, if I go really, really deep, there's a whole bunch of different bar magnets. And these bar magnets are going to be really important. Why? Next slide, please. So what I show on the left, where you have that sort of funny looking white thing, that funny looking white thing costs about $25 million. So it's quite expensive. It's not chump change. We need to get big grants from the government to be able to buy these machines. And these machines produce a super big magnetic field, many, many hundreds of thousands of times the Earth's magnetic field. So you can think about that white thing on the left as a super big magnet. It has a north pole and a south pole. And then what I'm going to do is I'm going to take my protein molecules, which are made up of very, very tiny magnets. Remember, I said that the nucleus of every atom is essentially a little bar magnet. And I'm going to take my tiny protein molecules, I'm going to put it in the big magnet. Now, if we move to the next picture right beside the large magnet, so you can see the N in blue on the top and the S in red on the bottom. Hopefully you can see that. That is the large magnet that I spent $25 million of your parents' tax money on. And then the other little magnet, that's the large one, but there's a little magnet there, which has just the red and the blue in between, the big blue and the big red. That's the little bar magnet associated with every atom of my protein molecule. And these little bar magnets want to align such that the south pole of the little tiny atomic bar magnet aligns with the north pole of the big magnet that cost me $25 million. And then what I'm going to do is I'm going to put energy into the system. I'm going to give the tiny little bar magnets that are associated with the atoms or the nuclei a kick, such that the blue now becomes close to the blue shown on the right-hand side. That's a high energy state. That's not very happy state. And so what happens over time is the state shown on the right goes to the state that is shown in the middle. Okay, and the question is, I put energy into these little bar magnets, and how can that tell us anything about the molecules in which these tiny atomic bar magnets are found? And the reason is because the rate at which the energy in the high energy state is dissipated, goes back to the equilibrium low energy state, is related to the relative positions of the atoms and how they change in time. So that if I measure how long it takes for every different atom, I can get information about the position of the other atoms with respect to it. I can get the structure, the three-dimensional structure and how it changes with respect to time. Think about it this way. Supposing you have a punching bag. Now, you know how punching bags work. You punch the punching bag. The punching bag absorbs your anger or your energy. It goes prone onto the floor, and then it comes back over time. Once you punch the punching bag, you've applied energy into it, and you can watch it come back. But there's a lot of different ways you can apply energy to a punching bag. You can apply one punch or 10 punches. You can apply the punches at different positions along the punching bag at different times, right? It can be very rapid punches or very slow punches. So there's a lot of different experiments that I, as a physicist, can develop, focusing on the transfer of energy this is magnetic energy that I'm transferring inside the nuclei of the atoms that in turn make up the protein molecules. I can watch how that energy is released, and that gives me information about the structure and the dynamics. Next slide, please. So I develop experiments using math and physics and apply them to molecules. And here you can see on the right is my molecule. Looks like a virus molecule in pink, and I'm giving the molecule a kick. Well, I don't really use my foot to give the molecule a kick. I use actually electromagnetic radiation that gives my molecule a little kick that allows these atoms to change their orientation in a big magnetic field. And I then watch how that change in orientation, which brings a north pole close to a north pole, is restored to a much more favorable north pole close to south pole. Next slide, please. I want to conclude by just giving you one example. These are examples of a molecule. In the upper right molecule, upper left-hand side molecule in pink, you can see the arms. The arms are the blue things, the blue parts of the molecule. And you can see that those arms are down by the side of the molecule. In contrast, in the lower depiction, you have the blue arms that are up. So think about it as your arms. Your arms can be up if you want to catch a football, or maybe they're down if you have to put your hand in your pants to take out your wallet and pay a bill. And clearly, to carry out a particular function, the arms have to be in the right place. Now, each one of these structures Each one of these ovals, whether it's a pink oval or a gray oval, is made up of 100 beads. And if one of the beads, only one of the beads is wrong, it's yellow instead of blue, for example, or pink instead of red, that's enough to give you a disease where the arms that should be up under certain conditions are now always down, or vice versa. And that means the molecules don't function, and that leads to neurodegeneration, and that leads to death. Just one mistake out of 800. And so one of the things that I do is to try to understand why it is that that one mistake can cause such a devastating disease, and how can we fix that one mistake so that the arms know where they're supposed to be at the right time so that the molecule can do its function. Next slide, please. So let me just conclude by saying that science is super creative. You have to learn the rules, and it takes a long time to learn the rules, but playing the game is super rewarding because I get to do whatever I want. I get to come up with new ideas that will make my molecules reveal their inner secrets so I can understand how they work. Don't get discouraged. Remember that science is easier than getting tackled by a 300-pound super big person in football. And always work hard and play hard and believe in yourselves. Thank you very much.
Thank you so much, Lewis, and, for such a deep dive on so many cool things. Again, this is going to be on our YouTube channel, so people can check this out 3 weeks, 3 years from now. Lots of opportunity to keep the learning going. And we've already got a bevy of great questions. So you talked about proteins a lot in that program, a literally deep dive in on them. What tools do you use to look in on proteins? Like, what's the actual apparatus you'd use to look at something like that? Do you have something like that? Can you tell us a little more about it?
Right. So those are those magnets that I showed you, or that picture of the spectrometer that I showed you. So I use a technique called nuclear magnetic resonance spectroscopy. So that's actually a mouthful. Most of you probably won't have heard of NMR spectroscopy, but some of you may even have had MRIs. An MRI is imaging a foot or a head or a part of your body. It's the equivalent of an MRI, but I image molecules. Okay? Rather than looking at feet or heads or limbs, I look at molecules. So think about it as an MRI just specialized for tiny little molecules. Now, my tiny little molecules are in solution. They're in a solution. So think about water sprinkled with a little, or actually many molecules. I put those molecules in my magnetic field. This is a very big magnetic field that is very, very expensive. And then these atoms, or the nuclei are in atoms. The atoms are in the molecules that I'm looking at. They orient with respect to the magnetic field. And then I have a computer that tells how I want to apply energy, magnetic energy, into the system, which allows the orientations of the little bar magnets, the atomic bar magnets, to change. And I design experiments by writing computer programs based on physics, and physics called quantum mechanics, which is the way that we can understand how very tiny things interact with fields, like a magnetic field or an electric field. Understanding that allows me to design experiments where I can see how the various atoms move around each other. But it all comes from a big piece of equipment called an NMR, nuclear magnetic resonance spectrometer, which is sort of like an MRI. MRIs have big magnets, and they apply energy, in this case, into a human body in a particular region, so that, in that case, imaging of an organ is possible, or a limb. In my case, I image tiny things, tiny little molecules.
Super cool. Thank you so much, Lewis. All right, we've got a query about whether AI is of any use in understanding proteins. And I love this question because when we talk about AI and a lot of things, I actually guide it back to how we use it for proteins. But could you tell us a little bit more about that?
Yes. So AI is super important for understanding the shapes of protein molecules. So many years ago, people didn't have AI, and they generated the shapes of a lot of different proteins. So we have a database which has, literally hundreds of thousands of protein structures. And you can train computer programs on that so that they can, given a new sequence, and what do I mean by a sequence of protein? Of a protein, I mean the order of the colors of the beads. Remember, I showed you this bead on a string. So if we have a string and we have a yellow bead and then 2 blue beads and then a black bead, etc., and we have another protein which would have a different order of colors, knowing the order of colors, there is now an approach which allows one to get an overall 3-dimensional structure. And that's wonderful for me because it means that I don't have to worry anymore about getting these structures. I can rely on computation to get them, which means I can focus on what I'm really interested in, which AI can't do now and won't be able to do for a long time, which is understand how these structures change in time. I can focus my energy having a starting point, knowing that at time 0, my molecule looks like this. How does it evolve in time as it interacts with other molecules, as it swims about in solution? Because remember, these protein molecules are not static entities. They're not at absolute 0. They're not at a super cold temperature. You and I are not super cooled. We're at a, little bit hotter than room temperature. So our molecules are moving about. Not only are they diffusing in the cell to the extent that they can, because remember, the cell is super densely packed with molecules, but they're jiggling and jaggling. Various parts of the protein molecule are moving with respect to each other. And that's what I want to understand, because it's that motion which is often really important for function. And when that motion is changed, leads to misfunction and disease. So now that I have a good guess about how the molecule might look like at time 0, I can begin to focus my experiments in a much more focused way, in a much more detailed way on understanding how the shape of the molecule, how the structure of the molecule evolves in time, and how that evolution leads to biological function.
Very cool. Louis, thanks for the answer, man. This is great. We got time for a few more questions together. So one of our ones from our classes is, do we know all the proteins? So you talked about all the different kinds of proteins. You showed a bunch of examples of them in 2D. Do we know every protein that exists, or are there many more to find?
That's an interesting question. Certainly we know a lot of them, and we have the structures of now many proteins. We can predict them very, very, very rapidly. But proteins interact. They're not entities by themselves. They interact with other proteins. There's a whole network of interactions. A interacts with B, that then interacts with C, but only if A is bound to B. So A may not interact with C unless it's bound to B, but if A is bound to B and C, maybe it doesn't interact with E, but it interacts with F. Understanding the structures of these complexes, understanding what complexes are involved, understanding all of the molecular players of these complexes is an active area of research, and understanding how the molecules of life interact with each other and at what times during the lifetime of a cell do they interact, and what are the cues, what are the signals which cause things to interact or not interact, and how do those signals change as a function of mutations that lead to disease. These are all things that have to be explored in a lot more detail. So if you're asking me whether I'm going to be unemployed in a few years, the answer is an emphatic no.
Good.
And so will you be if you want to go into science as well.
It's a golden time for science. And I mean, we highlight this in all our programs, no matter what the specific discipline might be, but I'm happy to hear it for our student audience. Louis, with one final question before we wrap up and say farewell, because time always flies when you're doing these broadcasts. Can we make our own proteins? Like if you set out to make a new protein, is that possible? And what would it be used for? Could we use it for something? What's the deal?
That's a great question. Well, I want to refer you to the Nobel Prize in Chemistry 2024. David Baker got the Nobel Prize for precisely those answering that question. Indeed, he was able to, through computation, through important experiments that he did, show that, in fact, one can design all sorts of different proteins. You can have designer proteins that may eat plastics in the ocean so that we can clean up oceans. You can have designer proteins, new antibodies that can target cancer cells and get rid of them. You can have designer proteins new enzymes that may function more efficiently. All sorts of different proteins can now be designed. You can have proteins, for example, that are in the body that say, okay, I detect something that shouldn't be here that may aggregate. So I'm going to bind to it and basically get rid of it, clear it from the bloodstream. So you can design a whole bunch of different types of proteins that carry out a whole bunch of different functions, all from biomedical applications to industrial applications. Very important that we design enzymes that carry out functions at very high temperatures where chemical reactions can occur in a more efficient way. That lowers the cost of the ultimate product. So there's just so many different possibilities for designing of proteins that will ultimately impact society in so many different ways. The answer to that question is a big, big yes, and it was recognized by the Nobel Prize in Chemistry in 2024.
What a great segue. Lewis, this has been so much fun as always. Thank you so much for making this work, making the time for us in this Gardner series. It's been a thrill every single year to have you on. And since you'll be in the job in years to come, we'll just keep having you back on so we can highlight all the newest, latest, greatest research.
I look forward to it. Thank you so much, Jesse.
Amazing. Well, for now, folks, if you're joining in on YouTube live or afterwards, thank you so much for tuning in. I hope your questions got answered. And if you have any more, you can always email us at exploringbytheseatofyourpants as well. But Lewis, we'll say farewell for now. Bye, everyone, and have a wonderful rest of